Hello, my name is Joanne. And we're going to be talking about the formation, docking, and fusion of vesicles. There are two main pathways for vesicle transport. The first one is secretory, and the second one is endocytic. The secretory pathway is where synthesized proteins or lipids are secreted outside of the cell, and the endocytic pathway is where extracellular molecules come into the cell and are degraded. The topics of discussion for today are vesicle function, vesicle formation, vesicle docking, vesicle fusion, endocytosis, and exocytosis. Vesicle formation. The vesicles are coated with a protein called clathrin, which bud off of the Golgi complex. This budding is done for secretion purposes involving the cell membrane. This secretion process is known as endocytosis. As the cell membrane, each vesicle starts off as a clathrin-coated pit. A small protein known as dynamin forms a ring around the neck of the vesicle and pinches off the rest of the membrane. The neck will eventually be constricted to a breaking point. This breaking point will cause the vesicle to be released. The clathrin binds to a second class of vesicle coat proteins known as adaptins. These will choose the cargo that is loaded up into the vesicle. These adaptins are bound to cargo receptors that contain transport signals. They allow for the capturing of molecules by trapping the receptors below the neck of the vesicle. All transport vesicles have different protein coats and different adaptins for different membranes. These variations also enhance the difference in receptors. All these differences allow for vesicle specificity. COP-coated vesicles transport cargo between the ER and the Golgi. These vesicles get from one location to the next by using walking proteins. Walking proteins physically walk across microtubules while they carry vesicles. Once the vesicle has reached the target organelle, it will recognize the membrane and dock and fuse. When we dock and fuse, we use specialized proteins. These specialized proteins are known as Rab proteins that are stuck on the vesicle membrane. Rab proteins are recognized and bound by tethering proteins that are present on the destination membrane. This binding of Rab and tethering proteins is known as docking. There is an additional recognition using snare proteins. These proteins stabilize the docking process by adding that second layer of anchoring. Membranes must fuse, connecting the vessel to the target. Then there must be a releasing of those vesicle contents. Once this occurs, the vesicle membrane becomes the target membrane. This is known as fusion.